Welcome everybody to this webinar. Today we are going to introduce our technology for the bioengineering of human tissues. This is an overview of some of the models that we have created, and subsequently this can be followed up by more in-depth presentations on specific models. For example, whether that be the skin model or the intestinal model. So for today, you just activate my laser pointer. Okay, there we are. Okay, so in our laboratory at Durham University, we have been working for many years investigating how the microenvironment in which cells grow in a petri dish can influence the outcome of experiments, and this is an effect through the action of various parameters on the cells growing in the dish. So the cell on the slide is affected by lots of different factors, as you can see, surrounding it, including the makeup of the medium, physical factors, oxygen, tension, three-dimensional space, and interactions with other cells, etc. And all this takes place over time. So that environment we can control, but also it can, as I say, affect the cell's behavior. So in vivo, of course, cells live in a natural environment, and they do so with their neighboring cells and the extracellular matrix, constituting a tissue composed of multiple cell types. In vitro, we often break these connections between cells. We seed them most often into sterile vessels, whether they be plastic or glass, and the cells adapt to that foreign environment. They change their shape and form and architecture, and as a consequence, they re-engineer their internal cytoskeleton. They become polarized, and this can affect gene transcription, protein translation, and function. So the function of the now adapted flattened cell is different to its native three-dimensional counterpart. So we're interested in developing technology that enables us to grow cells in a more natural environment. So we're trying to recreate that three-dimensional architecture to provide an opportunity for the cells to grow with each other in a three-dimensional syncytium with extracellular matrix and alternative cell types. So we're getting closer to what we find in a tissue. If we can do that, we can achieve more genuine 3D tissue architecture, which is more representative. We see enhanced structure and function, and these assays then, or these models rather, can provide us with useful assays with improved predictive accuracy. So this is what we've been working on, and we've now moved on to what we call bioengineering of human tissues using a technology that has enabled us to achieve this growth in 3D, which I will introduce shortly. So what we have on the right here are examples of tissue models that we have constructed using the technology I'm going to introduce to you. They follow anatomical and physiological principles in that their structure is related to their function. They are grown on a physical scaffold support, which I will mention shortly. We have also developed unique plasticware to enable this process to happen. And each of the models have defined standard operating procedures and methodologies. They're highly characterized and they're robust and reproducible. We compare the models directly with real tissue, or both those of healthy and diseased tissues. For many of the models, we have extensive application data sets and publications. You will see as we go through the presentation, there are multiple endpoint assay options that users can choose. And we also work very closely with industry, understanding the needs of our commercial clients and developing custom solutions where necessary. So this is important because such technology can advance research and discovery, improve efficiency, saving time and money, and also reduce and replace use of animals in research. So a bit of fundamental basic underpinning research here. So this is the technology that we developed in Durham, and it comprises of a porous polystyrene membrane. You can see this bottom left image. So that's it, edge on, and the middle image is high magnification. So it's 90% porous, meaning 90% space, only 10% polystyrene. So effectively what we've done is change the geometry of polystyrene. So cells no longer grow on the flat surface, as I mentioned, and adapt. They now enter and grow on top of this material. They no longer flatten, and they form connections with their neighboring cells and grow in 3 dimensions, and they produce their own extracellular matrix. And that's the basis of this technology. So this membrane, this porous membrane, acts as the catalyst upon which we bioengineer our human tissues. So this technology has been fully commercialized, and it is available through Reprocell, and its brand name is Alvetex. It's been on the market now for a number of years. It has established itself as the market-leading scaffold technology. There's over 150 independent publications from our customers that have bought, acquired, and used and published on this technology. It is available as multi-well plates, as you'll see, and also well inserts. So 6, 12, 24, for example. But it's beyond growing cells in 3D. It's now using this to bioengineer human tissue, which is the focus of today. And to do that, we also develop, design, and build our own plasticware. Because when we're growing tissue models in vitro, we have to be mindful that the demands of those models and access to those models is different from standard cell culture ware. So we've designed and built our own well inserts that contain the Alvetex. We have these inserts which can be presented at different heights, and we have deep well petri dishes, multi-well plates, so we can have larger volumes of media because there's many more cells per unit volume of medium compared to a normal culture. So one has to think about maintaining your cultures, especially over longer periods of time. And we've also designed our inserts to be compatible with some instrumentation which is used in the clinic. So this is a device here in this photograph, which is used to measure water loss from the surface of skin. And that device can also fit into our inserts very comfortably and operate as it would on real skin. So this technology is particularly useful for bioengineering of epithelial tissues because they are comprised anatomically of layers of different cell types. If we look at an epithelium in this schematic diagram, we see the epithelial cells resting on the basement membrane underpinned by fibroblasts and underlying tissues. So we're trying to use our technology to recapitulate this structure for different epithelial tissue types. And in this table, we see 4 simple examples. So intestine through to skin, and they each have specific structure function relationships. So in the intestine, of course, we want to enhance absorption and secretion. So we have a simple columnar epithelium. Whereas in the skin, it's protective, it acts as a waterproof barrier, etc. So we have a stratified keratinized. So using our technology, we simulate the ability to reproduce these structures in vitro. And this is what this presentation is about. And we'll start with skin as an example. So this is a stratified keratinized epithelium. So in simple terms, the skin comprises epidermis and dermis. These two compartments obviously interact with each other. The dermis is complex. It's comprised of multiple cell types, extracellular matrix, and provides a foundation to support the epidermis, which in itself is a complex differentiated epithelium comprising of a basal layer and stratum spinosum, granulosum, and corneum as the cells move towards the surface and differentiate. So how do we build this in our Alvetex constructs? So we take our membrane in the insert. We use 12 well format primarily for this, but we have done it in all the other formats as well. We seed into the insert containing Alvetex. We seed dermal primary human fibroblasts, and we give them 21 to 28 days to grow. And they grow in 3D inside and on the surface of the insert. And this period of time is important because during this period, the human fibroblasts are producing extracellular matrix. So we don't add any ECM to our models. It's all human derived. So you can see in the transmission electron micrograph here, a single fibroblast, and it's surrounded by these collagen fibrils, and these are all derived from fibroblast. And it's not just collagen. We know from proteomic analysis of ECM extracted from these models that we see a whole range of different proteins and their isoforms that one would expect to see in the dermis, including signaling molecules, etc. In the immunostaining on the left, we see examples of collagen 1, 3, and fibronectin increasing in expression over time. And we can also use biochemical assays to see the accumulation of total collagen, in this case, over 5 weeks. So we're creating this foundation using the Alvetex scaffold, upon which we then seed the epidermal keratinocytes. We raise to the air liquid interface, and then we see the stratification of the epithelium and the formation of the individual sublayers, as I mentioned before. So the basale, spinosum, granulosum, and corneum. This is a robust and reproducible process. It enables us to do recovery studies because we maintain these models for several weeks. And that enables us then to do things like wounding, etc. And I'll show you some examples. This is, as I say, reproducible. These are different batches of cells used to make models. And the top model there you can see is over 1 centimeter in diameter. These models are highly characterized. So this is immunostaining for proteins expressed in real skin. So we compare our model against real tissue, and we do so for a variety of different markers, as shown in this slide. And all this is published incidentally, so you can look at this at your leisure later. Excuse me. We also perform electron microscopy, both scanning and transmission, and we see the ultrastructure of the cell model. So we see here the hemidesmosomes, the desmosomes, the corneodesmosomes. These are junctional complexes found in the skin, the mellobodies, etc. So all of that structure is as it should be. So once we have this skin platform, we can then use it for lots of different applications, and we can enhance its complexity by introducing additional cell populations. We'll focus on examples such as pigmented skin, neurosensory and immune as well. So once we have this system, we can use it for a variety of different purposes. And we have worked with skin care companies developing consumable products such as Procter & Gamble. We've published a lot of our data together, and you can read about this in our papers. A lot of this technology can be used to explore things such as wounding, the effect of UV radiation on pigmented skin, pollution, etc. So I'll take you through some examples. So aging skin. So how do we produce our models of aged skin? Well, we can do one of two ways. The first way is to utilize cells from older donors. So what we see here in these images, on the left is a young skin model. So that's neonatal fibroblasts and keratinocytes, compared to, on the right, an aging skin model. So these are fibroblasts derived from a 50-year-old, and you notice immediately the thinning of the epidermis, and there's also less collagen in the dermal compartment. So that's recapitulating a couple of the basic phenotypic differences that one sees as one ages. An alternative strategy is to introduce into a young model an increasing percentage of senescent cells. So we render our fibroblasts senescent using X-ray radiation. We incorporate them into the dermal compartment, and we see then the effect of the bystander effect known as SASP. So the effect from the senescent cells, which influences surrounding tissues, mimicking the aging phenotype. We see a thinning of the epidermis, as you can see along the top. So that's two strategies by which we can develop an aging model. And of course, with this one, the senescent one, we can use that for targeting the development of senolytics and senomorphic treatments. So here's an example of where we've incorporated an additional cell type. This is the melanocyte. So melanocytes produce melanin, and they transfer those melanosomes via their dendrites to the adjacent keratinocytes. The melanosomes then form a supranuclear cap above the nucleus in real skin. So you can see that very nicely here. So this is a supranuclear cap of melanosomes above the nucleus, protecting the nucleic acid from UV radiation. And one of the first questions we wanted to know was whether in our model we saw supranuclear caps forming. So here's a very nice example of a dendritic melanocyte in the stratum basale of our skin model. And you'll see very nicely examples of supranuclear caps as well. And we've looked at this in detail. Again, this is published work using transmission electron microscopy. So we can create pigmented models from donors of different ethnicities. We create a skin tone palette, which is a valuable research tool for exploring how skin of different skin tones reacts to different conditions, and we can learn a lot from these particular models. And I'll give you some examples. Before I do, I just want to demonstrate the importance of co-culture. So what's really interesting is if we create an epidermal only model and incorporate melanocytes, so this is what you see here in panel C, see the melanocytes, but there are no supranuclear caps. Only if we have the dermal compartment present do you get the correct organization in the tissue model. And that demonstrates nicely that there must be some interaction between the dermis and the epidermis in order for the correct anatomy of the epidermis to form, which is not surprising, and one would expect that to happen, as certainly different cell populations signal to each other. So if we take our pigmented skin models, we can expose them to various molecules, and there are products on the market which are designed to lighten skin pigmentation. So kojic acid is a well-known ingredient of such products. We can incorporate that into our cell culture media, expose it to the models, and we see lightening of the skin in a concentration-dependent manner. We can record from the models. So these are actually the gross photography of the models here. You can see in this photograph, you see them lightening, but we can use the device called the color catch, and I'll show you that on the next slide. It's a handheld instrument which you put onto the surface of skin, and it will measure the melanin index and the ITA, which are references of skin tone. So you can quantify it, and you can see that very nicely as melanin index decreases as kojic acid increases. So this is the color catch here. This is the device which sits on top of the skin models, as I mentioned. In this slide, we're showing how we can expose our skin models to UV irradiation. So we have the Biosun irradiator, which delivers specific amounts of UVA and UVB radiation. This, we can put our models into here, deliver the UV, and then we see a darkening of our model. So essentially we're giving it a suntan using this system. And we can again measure that using the color catch. So this is valuable because then we can explore the use of, say, sunscreens. So this is an example where we are applying a sunscreen to the surface of the model. So these models, you can see this is the surface of the skin model here. They are actually quite robust, and you can interact with the surface of the skin very well. And I'll give some more examples shortly. So this operator here is applying the sunscreen, and we're then exposing models to UV radiation with and without sunscreen. So non-pigmented models with relatively low energy, you see blistering and sunburn cells forming, whereas in the presence of the sunscreen, SPF 50, you get total protection. In the pigmented model, we have to turn the power up to get a burn, and then we get protection with the sunscreen. In more recent data, we've also shown that in pigmented models, if we produce models with neonatal or middle-aged or old-aged cells, and we incorporate the melanocytes, we also see a lightening of the skin and a change in the distribution of melanin in the models. And this is interesting because this could simulate age spots, which appear, of course, on individuals as they age. You'll notice them on the back of people's hands, for example, exposed particularly to light. And this is something which we're currently developing further. So that's very unique to our work. So this is another way we can interact with the surface of the skin. So this is an exfoliation technique we've developed and published on with P&G. So we've taken our human skin models, we've put tape strips onto the surface and removed the upper layer of cells. And if we repeat this, we can get down to what's called the glistening layer. That's about 10 tape strips. And what's nice about this is that if you then put those models back, you see them then come back and grow and recover. So this is an example of how you can interact with the system, challenge it with an injury, or add a active and then study its effect over time in a live culture environment, which is clearly beneficial, and it then can produce data which can help with the design of clinical trials, etc. So we can use this particular example as a surrogate for shaving, where it's known that a razor will remove the upper layers of the stratum corneum. And we see here in these data how as the number of tape strips increases, the tool, so this is the trans epidermal water loss, increases, and we measure that using our vipometer, that device I showed you, which is fitted into the inserts. And we see how the tool recovers again over time. We see a cellular response in our models. This is a enhanced cell proliferation in response to the tape strip, and we again see recovery. So another way, of course, is to actually put a blade across the model, and we can do that, simulating a shaving response, and we can see how tool increases with a razor, and also we can collect material from the razor which has been removed and analyze it. We can demonstrate very nicely the difference between dry shaving and shaving with a shaving gel or a foam applied to the surface of the model. And we see how these products protect the skin as it should during a shave. We can mimic all of this using our technology. Sometimes there's damage to the surface of the skin, so we're here showing an example of a superficial injury. This is a wound, and we can mimic this in various ways. This is actually using, in this case, a drag of a scalpel blade across the surface, and you can see the vertical line in these models. And because these are live models and we can maintain them, we can image them on a day-by-day basis. And we've actually just also acquired the VisioScan technology, which is used in the cosmetics industry to apply to our models. So we can use that as an output as well. So these are the scratches you see on the skin. They're superficial. You can see that very nicely. Break in the epidermis, and we see then this is an example of how the barrier has been penetrated. So this is a lucifer dye penetrating now into the tissue underneath. The tool increases and then recovers as the model then heals over time. And you can follow this. This is a partial cut over 7 days. And you can see how at first you've got the cut, it broadens because it contracts. And then we see the flood of cells migrating into the site of injury and then recovery. We can look at this in more detail using scanning electron microscopy, so you can see inside the wound, cells and the ECM. Another strategy is to burn the model. So we've got this rig set up here where we have a branding iron set at different temperatures. So we particularly use 70 and 100 degrees. We bring that onto the surface of the model. We brand it, we burn it. So you can see very nicely this particular tip has been cut for a rectangular shape, but we have other shapes as well. And we can brand the surface of the skin. In the wound area of the burn, you'll see that the cells die off. And then over time, the adjacent area surrounding the wound, the cells begin to migrate into this space as the model heals itself. And you can see nice examples of, for example, here keratin 14 expression being driven and increasing as the cells migrate into that space. We can also incorporate into our model immune cells. So in this study, we were interested in the exposure of the human skin to particulate pollution from diesel fumes. So we incorporated Langerhans cells into the epidermis. So these were derived from the MUTS3 lineage and then incorporated into the skin. You see the green cells here. This is a marker of the Langerhans cells. So they're in the epidermis. And then we expose the models to the diesel particulates. Stratum corneum is very good at keeping the particles out of the skin, but it doesn't stop the chemicals on the particles which move into the skin and cause local pro-inflammatory responses. We can monitor that by gene expression or by cytokine analysis. as you see in these data here. So sensitive techniques can be used to study the effect of pro-inflammatory activators on the surface of the skin. And more recently, we've now introduced macrophages as well into our system. So these are derived from the THP1 lineage. These also carry the NF-kappa B reporter, and I'll talk more about these cells in our intestinal model shortly. So that's another element of our skin immune system we've built in. Also incorporated neurons into the model. So you can see very nicely these sensory neurons beneath the epidermis. Their free nerve endings reach up into the epidermis. And this is valuable because we can test now products which may well have a sensorial effect on the surface of skin. When a user puts on, say, a particular cosmetic, it might give a mild burning sensation. So we can assess whether that's going to be possible. In this example, these data here, what they show is the addition of capsaicin to the surface of our models results in the release of neuropeptides, which we can then assay in our models, and that equates to the effect that we see. So you could use this system as a screening tool in advance of clinical trials. So that's skin. So we have a significant body of data now and publications in relation to the development of the skin model and now its application. Let's turn our attention to the other model for today. That's the intestinal mucosa. So this is a simple columnar epithelium, and this one is built using a cell line. So CACO2, as many of you I'm sure will know, is used heavily in industry and academia to study the transit of molecules across the intestinal wall, most often in a transwell. And it has limitations. So it has a very high resistance, it's overly simplified, and the anatomy of these cells bears no resemblance really to, or very little resemblance to, the real tissue. So we hypothesized if we took CACO2 and we put it into our model on top of fibroblasts, it would change the anatomy and the physiology of these cells. So indeed, that's what happens. The CACO2 cells now become prominently columnar. They have a prominent brush border, and they are polarized, as their native counterparts would be. So you can see them sitting nicely on top of the fibroblasts. Very different to their 2D counterparts in transwells. Maintain these for many weeks, and you see up to, in this example, 5 weeks. So nice and reproducible. Lots of characterization data again. So here are the fibroblasts and the top scanning electron micrograph images, and now we've got the CACOs on the top, the bottom, cobblestone appearance at the surface of the cells, and at high mag you can see the glycocalyx and microvilli. These are transmission electron micrographs of the microvilli, tight junctional complexes at the apical end and the basement membrane at the basal end. We do a comparison of in vitro versus in vivo, just like before, so model versus real tissue for a variety of different markers. Again, the expression patterns are as we would anticipate. These models can be encapsulated between 2 chambers in a nushing setup. We can then measure the electrophysiology of these models. We can study secretion, absorption responses. We can challenge, or we can add drugs, etc. What we've done with this model in particular is used it as a basis of a disease model of IBD, so inflammatory bowel disease. So that's ulcerative colitis and Crohn's. This is an inflammatory disorder affecting a significant proportion of our population. And it's characterized by immune cell infiltration, leaky gut, and changes to the extracellular matrix. So these 3 hallmarks, the inflammatory response, the remodeling of ECM, and the leaky gut, they're 3 hallmarks that we can simultaneously observe in our model. And I will share that with you now. So first of all, we incorporate into our intestinal model the immune cells, and we use, again, this THP1 lineage. So these are macrophages. You can see them very nicely within the model. So see them, these larger round cells, but you can confirm their identity by CD antigens in the immunostaining. So beneath the epithelium, these are the CACO2s, fibroblasts in here. We've got the immune cells as well, just as depicted in this schematic. And we've also done some comparisons of the model with the disease tissue also. So this particular lineage carries this NF-kappa B reporter, which is quite handy because it then enables us to do a readout by a simple plate assay. You get a color change effect when the cells are stimulated. So we can stimulate the cells in the model using LPS, and we then get a cytokine storm of pro-inflammatory markers being released, which are typical of Crohn's and colitis. So we can measure all of that using multiplex analysis on arrays, having collected the media. We also see very nicely that this inflammatory response is also having an impact on paracellular permeability, mimicking the leaky gut syndrome. So the tear levels of the model decrease, the passage of fluorescent dyes and other molecular markers such as certain dextran molecules we've used also increase. So you can figure out the permeability using those methods. And very interestingly, there are also changes to the extracellular matrix within the model. So we noticed in our model MMP9 levels being enhanced beneath the basement membrane. So we went back to the pathology tissue, and we noted and recorded for the first time the observation of MMP9 expression being very high at this area in the tissue. So that's an example of how studying these bioengineered human models can also then impact on our understanding of what's going on in the pathology of the disease. So summary slide for gut, and just very quickly, it shows very nicely here the incorporation of mucus producing cells into the epithelium as well. So we've also got that as an option. And it's a very versatile system. I mentioned at the beginning, endpoint assays. So here are a list of endpoint assays that this technology can be used for. Luminex for expression analyses, protein, electrophysiology, paracellular permeability, and standard techniques. So just very quickly to wrap up, I'll introduce two more technologies. This is oral mucosa, 2 slides on each. So this is a stratified epithelia, but it's non-keratinized. So this is the inside of the mouth, so it's a wet epithelium. You can see very clearly here, stratified cell epithelium sitting on this bed of Alvetex with fibroblasts. Again, we've got very detailed analysis of the anatomy. This is the ultrastructure showing the junctional complexes, the basement membrane, the structure of the cells, the expression of various markers between the model and real tissue, all as it should be. And the last example is the human nasal mucosal system that we've generated. This one's slightly different. This is a pseudo-stratified ciliated epithelium. See the cilia very prominently here. This is a goblet cell. These are the fibroblasts, and this is the tight junction along here, interface between the two compartments. These produce mucus. They move the mucus around in the culture media, and you can see all of this happening. That's an opportunity for respiratory disorder investigations and also drug delivery, potentially delivery of vaccines via nebulizers. Again, lots of characterization. This is just some data showing the ultrastructure of the cilia and the basement membrane. So cilia, 9+2 centriole arrangement, very classical, and then basement membrane here. So again, the anatomy of the model is replicating that what we see in reality in the real tissue. Okay, so final slide, key messages. Bioengineering human tissues in vitro. So we've introduced some models, some examples. So the key messages are that these models are built on a physical support, this Alvetex membrane I introduced to you. We also build and design our own bespoke plasticware to go with this. Each of these models is refined and has an optimized standard operating procedure behind it. They're robust and reproducible. There's published papers, and their in-depth characterization is, as I've shown you, again, real tissue versus the model, and also in disease circumstances. You can use these systems for acute chronic exposures and recovery as well over time. That's a real advantage of this technology. Some of these models have really extensive data sets and application notes behind them. I think from the presentation, you probably gathered that there are multiple endpoint analyses. All the things you can do with real tissue, pretty much you can do with this. So molecular cell biology, gene expression, protein analysis, electrophysiology in some circumstances, etc. And the last point to make is that we work heavily with clients, both in industry, collaborate with academic groups. We help get you started. You can obviously buy the technology, the Alvetex, and develop your own model. Or if you're in industry, perhaps you might want to work closely with Reprocell as a CRO to enable the access to the technology so you can test your own formulations, your new products on our models, and we will obviously do that for you and share the data.